I know you are able to hear me. Just wait and call. Hi, good morning. Can you tell me the screen is visible to you all? Just tell me whether the screen is okay. Okay. The sound is there. You are able to hear my sound. Audio is clear now. Okay. After 1-2 minutes, we will start with this. Okay. Good morning, everybody. Hope everyone is fine there. You all are following your COVID appropriate behavior. So today we're going to start your lecture about neuromuscular. We have completed your nerve physiology. So there we have seen all the things about nerve. Now we are going proceeding to your next lecture. And in today's lecture, we will discuss something about your neuromuscular junction and few things about your skeletal muscles. Okay. I think Dr. Ravi may have taught you a few things about that. So it would be a kind of a revision also, but this would be a former class, and we will cover all the aspects of neuromuscular junctions and your skeletal muscles there. So let's start the lecture. So before starting about the neuromuscular junction, we would first discuss in brief about some anatomical aspects of your spinal cord. So if you see a cross section of your spinal cord, so then you will see that in the spinal cord, inside of the spinal cord there is a H-shaped gray matter, and on the outside it is surrounded by white matter. So we know that white matter is made up of myelinated axon, and gray matter is made up of unmyelinated cell bodies there, or soma of the neurons. So inside of this cross section, the circular cross section of spinal cord, you would be having H-shaped gray matter there. In the anterior, you would be seeing that there is a anterior median fissure, and on the posterior there is a posterior median fissure is there. And this gray shaped this gray matter, which is in the form of S shape, it has two horns. One is your on the front you have ventral horn, or and the back you have a dorsal horn. And these dorsal and ventral horns are connected by the roots. And on the anterior you have ventral roots, and on the posterior you have a dorsal root. Now, the importance of knowing thing is that on the dorsal root, it is through this dorsal root all the sensory nerves are entering into the spinal cord, and it is through this ventral root all the motor nerves are coming out of your spinal cord. You will see here in this diagram also the ventral roots basically contain your motor neurons, which are innervating your muscles, and all the signals which are coming out of your spinal cord, they are coming out of this motor neuron. And similarly, in the posterior root or dorsal root, they receive all the sensory nerves which are coming from the periphery to the spinal cord. Okay, and it is through this route all the sensory information are entering into the spinal cord. So what I want to say, that remember that ventral root is motor in character and the dorsal root is sensory in character. And this concept is known as Bell-Magendie law. Got it? Bell-Magendie law. So two things are clear from this, that ventral is motor. It is through this that motor neurons are coming, and the dorsal root is sensory. All the sensory nerves which are entering into the spinal cord, they are entering through the dorsal root of spinal cord. Okay. Now coming to the today's topic of discussion, we know that from the ventral root of a spinal cord, large myelinated nerve fibers are coming, and these nerve fibers which are coming, they divide into multiple branches once they enter into the belly of the muscle. And these multiple branches of the motor nerve, which is coming from the ventral root of spinal cord, they are innervating the different types of muscle fibers which are present in the muscle belly. Now this contact between the nerve fiber and the muscle, we call them as neuromuscular junction. So remember, skeletal muscle fibers are innervated by large myelinated nerve fiber which are coming from the ventral root of spinal cord. So neuromuscular junction, what it is? It is a junction or point of contact between the large myelinated nerve fibers which are coming from the ventral root of spinal cord and the muscle fibers. So this is your neuromuscular junction. Now this axon terminal of the nerve fiber, they imaginate over the surface of the muscle fibers. But imagination doesn't mean penetration. Got it? They are just imaginating the surface of muscle fiber, and they lie on the outside of the plasma membrane of the muscle fiber. So these nerve fiber, they imaginate the surface of muscle fiber, and this entire complex of your axon and the surface of muscle fiber, which is making contact with this axon. This entire complex is known as motor end plate, or otherwise we can call as neuromuscular junction. Okay. This motor end plate is surrounded by one or two Schwann cells, and the purpose of the Schwann cell is to insulate this structure of motor end plate from the surrounding extracellular fluid. So if you see the neuromuscular junction, this neuromuscular junction has two components. One component is coming from the nerve, which we call as presynaptic structure, and one component is the membrane of the muscle to which this nerve is making the contact. Okay, so this part is we call as muscle part, and it is a postsynaptic structure. So in a neuromuscular junction, you have two structure. One is your presynaptic structure and another is your postsynaptic structure. Presynaptic structure is the axon, which is making contact with the muscle fiber, and postsynaptic structure is the membrane of the muscle, which is making contact with that axon. So what is the role of neuromuscular junction? We all know the role of the neuromuscular junction is communication. This transmission of the impulses which is there in the nerve fiber to the muscle fiber. We know that the muscle fiber are innervated by the nerves, and the signal for contraction to the muscle, they are coming, or the impulse, they are coming through the nerve. So what is happening there? We are stimulating a nerve, and in response to that stimulus, an action potential is created in the nerve, and this action potential is carried to the end of the nerve, and there they are releasing some neurotransmitter which are acting over the surface of muscle, and in response to that neurotransmitter which is acting over the muscle, there is opening of some ion channels, or sodium ion channels over the muscle, and because of that there's a depolarization. So this is the way the electrical signal is traveling or transmitted from the nerve to the muscle, and it's a type of chemical synapse. Why? When you will read about the synapse, synapse is what? It's a point of contact between one nerve to another nerve, or between nerve or muscle. It's a point of contact between nerve and other structure. So there you will read about that synapse could be classified into three categories. One is your electrical synapse, another is your chemical synapse, and another is your conjoint synapse. So in the electrical synapse, the transmission of signal is because of transmission of some ions from one cell to another cell. For example, gap junction is a very good example of your electrical synapse, where two cells are very close to each other and there is a movement of ion from one cell to another cell. The second category of synapse is your chemical synapse. Here, what is happening? Here, one neuron is releasing some neurotransmitter, and that neurotransmitter is acting over the adjacent neuron, and because of that, there is the opening of some ion channels, and because of opening of ion channels, there is entry of sodium, and there is a depolarization. So you see that here this transmission of electrical impulses is happening because of some chemical mediator or the neurotransmitter. So this type of synapse we call them as chemical synapse. Third category of synapse would be, we call them as conjoint synapse, where these kind of synapse have both the properties. That means they have electrical properties also and chemical properties also. So if you see the role of neuromuscular junction, the role of neuromuscular junction is the simple transmission of the electrical impulses from the nerve ending to the skeletal. Now, before discussing about all the things that is happening at the level of neuromuscular junction, we will first see some of the physiological anatomy of neuromuscular junction. So if you see what are the structures which are present at neuromuscular junction, the first structure is your axon terminal, which we call them as terminal button also. Then the membrane of axon terminal, which is making contact with the muscle, we call them as presynaptic membrane. Then you have postsynaptic membrane. It's the membrane of the muscle which is making in contact with the membrane of the neurons. Then the depression or imagination over the surface of the muscle, we call them as synaptic gutter. Then you have subneural cleft, and then synaptic cleft. We will read one by one about all them. First structure is a terminal button. So you see that the axon terminal is enlarged into a knob-like structure, and this knob-like structure fit in a shallow depression in the which is present in the underlying muscle structure. So this knob-like structure, which is making contact, which is fitting over the shallow depression of the muscle, we call them as axon. We call them as axon terminal. Then synaptic gutter. So the invaginated muscle membrane. This is known as synaptic gutter. And if you see the surface of the synaptic gutter, there you will see that there are numerous folds which are present in the synaptic gutter. These numerous folds which are present in the synaptic gutter, we call them as subneural clefts. And the purpose of this subneural cleft is that to increase the surface area of synaptic gutter. Got it? So that the neurotransmitter has more surface to act. More surface area means more number of receptors would be there. That means your neurotransmitter, which is released from the axon terminal, will be having larger area to act. Then synaptic space. Synaptic space is the space between presynaptic membrane and postsynaptic. This space is known as synaptic space. And normally it is 20 to 30 nanometer wide. Now coming to the side, acetylcholine. Now see, acetylcholine is the neurotransmitter which is released from the nerve endings, uh, uh, and this neurotransmitter act over the muscles, and in response to that, there's a depolarization of the muscle. So if you see the neurotransmitter acetylcholine, so these neurotransmitters are present into numerous small vesicles which is present at the end of axons. And these vesicles are formed by the Golgi apparatus. All these synaptic vesicles, they are formed by the Golgi apparatus, and through the exoplasmic flow, these synaptic vesicles, they move to the end of nerve terminal. Now, these neurotransmitter acetylcholine, they are synthesized in the cytoplasm, and then they are being absorbed rapidly into these synaptic vesicles. And it is said that each synaptic vesicle is having about 10,000 molecules of acetylcholine. And in one axon terminal, there is about 3 lakh synaptic vesicles which is present there. So about this neurotransmitter, what things you need to remember? Okay, they are present in synaptic vesicles. Synaptic vesicles are formed by Golgi apparatus. Got it? And they are being carried through exoplasmic flow to the end of nerve terminal. Second thing you have to remember, the acetylcholine is synthesized in the cytoplasm of the nerve, and then they are later absorbed in these synaptic vesicles, and then there are about, in each synaptic vesicle there are 10,000 molecules of acetylcholine, and about 3 lakh synaptic vesicles are present in the nerve terminal. Now, what are the sequence of event which is happening at the neuromuscular junction? So here we know that if you give a threshold stimulus to a nerve, then there will be generation of action potential, and this action potential will travel along the axon to reach the nerve terminal. Now, when these axon potential reaches to the nerve terminal, so at the nerve terminal you have voltage gated calcium channel. So because of the effect of depolarization, what will happen? These voltage gated calcium channel will open. Now, because of the because of opening of this voltage gated calcium channel, the calcium uptake will happen in the nerve terminal, and inside of the nerve terminal, the calcium concentration will increase by about 100 times. And because of this, what will happen? These synaptic vesicles, they will move to the ends of the axon. They will dock, and then these synaptic vesicles will fuse with the membrane, with presynaptic membrane, and there will be exocytosis. And because of this exocytosis, there is a release of neurotransmitter into the synaptic space. Now, how the exocytosis of this thing is happening? How calcium entry is causing exocytosis of these synaptic vesicles? Yeah, actually, what happened is whenever the calcium entry is there because of opening of voltage gated calcium channel, so this will lead to activation of calcium calmonine dependent protein kinase. And we know kinase means any enzyme which is going to cause phosphorylation. So because of activation of calcium calmonine dependent protein kinase, there is a phosphorylation of protein which we call them as synapsin. So this calcium dependent protein kinase will phosphorylate synapsin protein. Then normally what synapsin protein do? The normal function of synapsin protein is that this synapsin protein, because of this synapsin protein, these synaptic vesicles are attached to the cytoskeleton, which is present at the nerve terminal. Now, what happened? Because of the phosphorylation of synapsin protein, there will be a release of these vesicles from the cytoskeleton of the nerve terminal. And once they are released from the cytoskeletons of the nerve terminal, they will move to the active areas of the nerve terminal. There they will go. There is a docking of these vesicles, and then the membrane of the synaptic vesicle will fuse with the membrane of the nerve terminal. And because of this fusion, what happened? There is a release of the neurotransmitter which is present in the synaptic vesicle, and this release we call them as exocytosis of neurotransmitter. Now the neurotransmitter will come in the synaptic space, which is present between presynaptic membrane and postsynaptic membrane. Now this acetylcholine will travel the synaptic space, and they will act over the receptors which is present over the postsynaptic membrane. Now, what are the receptor which is present at the postsynaptic membrane? Acetylcholine, if you see, is a neurotransmitter, and it has two types of receptor. One type of receptor we call them as muscarinic receptor. And another type of receptor we call them as nicotinic receptor. Muscarinic receptors are mostly present in the ganglia, and nicotinic receptors are present in the at the neuromuscular junctions. So in the neuromuscular junction, the receptor over which the acetylcholine act, there is a receptor are nicotinic acetylcholine receptor. Now, if you see the character of this nicotinic acetylcholine receptor, they are ligand-gated ion channels. So we know that ligand-gated ion channels, they open or close in response to binding of some ligand. And here the ligand is acetylcholine. So what will happen? The acetylcholine will come, they will bind over the nicotinic acetylcholine receptor, and in response to this binding, there is an opening of acetylcholine receptor. There is a change in some configuration of this nicotinic acetylcholine receptor, and because of this change in the configuration, there is an increase in the permeability of motor end plate to the sodium ion. But these acetylcholine receptor, they are located near the mouth of subneural clefts. You have seen there is synaptic gutter, and in the synaptic gutter, the membrane is highly folded, which we call them as subneural cleft. So at the mouth of this subneural cleft, the acetylcholine receptor are located. And if you see the structure of acetylcholine, this structure of this nicotinic acetylcholine receptor, it has two alpha proteins, one beta, one delta, and one gamma protein. So in total, there are 5 subunits. 2 are alpha, 1 is beta, 1 is delta, and then 1 is gamma. So 5 subunits are there. And when 2 molecules of acetylcholine bind to these 2 alpha receptors, then there is a conformational changes. So for opening of nicotinic acetylcholine receptor, 2 molecules of acetylcholine must bind to the 2 alpha subunit. And whenever 2 molecule of acetylcholine will bind to these 2 alpha subunit, there will be change in the configuration of acetylcholine receptor, and because of this change in configuration, the permeability increases. As a result, large amount of sodium ion will influx. They will move from outside to inside of the cell. Now, what will happen? We all know that sodium ion is a positive ion. So whenever the sodium channel will open and there is the entry or influx of the sodium ion, there will be depolarization. So because of this depolarization, there will be depolarization will be there, and this depolarization which is happening at the end of the muscle membrane, we call them as end plate potential. Got it? And whether this end plate, remember that this end plate potential is not an action potential, it's a local or graded potential. Got it? Whether this end plate potential will cause action potential in muscle or not, it will depend upon what is the amplitude of this end plate potential. If the amplitude of end plate potential is sufficient to cause depolarization of 30 to 50 millivolt, then there will be generation of action potential in the membrane of muscle fiber. And once the action potential is created over the membrane of muscle fiber, it will spread throughout the membrane of muscle fiber. In this graph, you can see there is point A, B, and C. At point A, end plate potential was created, but this end plate the amplitude of this end plate potential was not able to reach to the threshold level. So here there is no generation of action potential. Similarly, if you see at this point C, there is also generation of end plate potential, but this end plate potential amplitude is not sufficient enough to take the membrane potential to threshold level. And since the membrane potential is not reaching to threshold level, so there is no generation of action potential. But if you see point B, here the amplitude of membrane potential was sufficient to carry the membrane potential to threshold level. As a result, there is a formation of an action potential. And once the action potential is created, it will travel throughout this membrane of muscle. Now, once the acetylcholine is released, then what is the fate of this acetylcholine? Remember that once the acetylcholine is released and once they have acted over the nicotinic acetylcholine receptor, they are destroyed in very few you can say in fraction of seconds. So most of these acetylcholines are destroyed because of an enzyme which is present in the synaptic cleft or synaptic space, which we call them as acetylcholine esterase. A small amount of acetylcholine may diffuse out of the synaptic space. So there is a two-way of destruction of acetylcholine. One is by acetylcholine esterase. It's an enzyme which is present in synaptic space. Second is because of some amount will diffuse out of the synaptic space. Now, this acetylcholine is broken down into two components, acetyl and choline. Choline is about most of the choline are they are reabsorbed by the nerve terminal, so that they could be reused for the formation of acetylcholine for further release. Now, these acetylcholines they are destroyed mostly to prevent repetitive muscle excitation. Got it? So they are destroyed in very fraction of second, and the purpose is that why acetylcholine is destroyed so fast. The reason is that to avoid repetitive muscle excitation, because still the acetylcholine is bound over the muscle, it will cause exciting. It will cause opening of the sodium ion channel, and it will cause depolarization, and because of that there could be a repetitive depolarization, and which can lead to repetitive sustained contraction of the muscle, and this sustained contraction of muscle we call them as tetany. So to avoid this condition, once the acetylcholine do its act, that is once they open the sodium channel and they have caused depolarization of muscle, they are destroyed in a fraction of second by enzyme acetylcholine esterase. Now, so we have seen about the structure of neuromuscular junction. We have seen what is the purpose of neuromuscular junction, and we have seen what are the events which is happening at the level of acetyl neuromuscular junction. That means how a nerve impulse is transmitted to muscle to cause its depolarization. Now, coming to two applied aspects, one is your myasthenia gravis, and second is your Lambert-Eaton syndrome. So there are these two diseases, myasthenia gravis and Lambert-Eaton syndrome, are autoimmune diseases. So you know the meaning of autoimmune diseases. So you know that in our body you have antibodies, and these antibodies they protect our body from any kind of external pathogens. And one of the very important characteristic of our immune system is that our immune system is able to differentiate between self and non-self. That means they are able to know that this antigen belongs to my body, or this antigen is a foreigner. So whenever an antigen which is coming from the outside, immune system is mounted against that antigen, and the antigens which are coming from, which are present in our body, they are not attacked by our immune system. But sometimes because of some reasons, there are many reasons which you may have read in your this chapter blood. So because of these reasons, this our body is not able to differentiate between self and non-self, and they are able to form antibodies against the against any body structure of our body. So the same thing is happening in myasthenia gravis. Here in myasthenia gravis, there is our body start forming antibodies against nicotinic acetylcholine receptor. So these antibodies they come and they bind over this nicotinic acetylcholine receptor. Now what happened, since most of the receptors are covered or bound by these antibodies, whatever the neurotransmitter which has been released at the nerve terminal, they are getting less number of receptor to bound, and since they are binding with less receptor, the amount of depolarization produced would be less. As a result, the muscle would not be contracting. As a result, there will be weakness in the muscle. Got it? So in myasthenia gravis, one of the very important property, one of the very important characteristic is that the patient will complain of muscle weakness. So muscle weakness is the reason for this muscle weakness is that most of the this nicotinic acetylcholine receptors are covered by these antibodies, and there are only few receptors which is left, and over these receptors, this neurotransmitter are acting, but because of these since very few receptors are bound with these antibodies, so very less amount of depolarization is produced. So one important characteristic is muscle weakness. Second important characteristic of this disease is that the patient will complain that the weakness increases on exertion. That means he will give a typical history that in the morning time I am fine, but as I start doing my daily activities, I start feeling weakness, and this weakness increases as I do my activities. And when I become when patient will feel fatigue, and once the patient will take a rest, and after taking rest, there will be some improvement in the muscular weakness. So here the weakness, there will be weakness on exertion, and on rest the patient will improve. Now why there is a weakness on exertion? See what is happening actually. The receptors are there. Some receptors are being covered by these antibodies. Okay. So there are less receptors, so the muscle, the patient may be having some weakness of the muscle. Now what happens as the fellow start doing the work, your nerve is being stimulated again and again, and because of this stimulation, the neurotransmitter are being released. Now when you are doing large amount of activities, then what will happen? There will be repetitive nerve stimulation, and there will be repetitive release of the neurotransmitter. Now your nerve has a limited capacity to release neurotransmitter. Got it? And if there is a repetitive stimulation, then what will happen? The rate of synthesis of neurotransmitter would be somewhat less as compared to the rate of release of neurotransmitter. Meaning that कि release ज़्यादा हो रहा है neurotransmitter का, synthesis कम हो रहा. तो क्या हो रहा है कि पहला प्रॉब्लम ये है कि receptors are bound by antibodies. दूसरा प्रॉब्लम ये है कि अगर the patient will do exertion, so what will happen कि the amount of neurotransmitter that is released, that will also decrease. Got it? Because जितना release हो रहा है उतना synthesis भी नहीं हो रहा. So on exertion there will be two problems. One problem will be that some amount of receptor would be covered by the antibody, and another problem will be that now the neurotransmitter which was released previously, that has also decreased. तो on exertion में दो परेशानी है. एक तो न्यूरो antibodies are covered by these nicotinic acetylcholine receptors are covered by antibody. दूसरा प्रॉब्लम ये है कि आपका जो न्यूरोट्रांसमीटर रिलीज हो रहा है वो भी रिड्यूस हो रहा है. So that's why it is said that there will be less release of neurotransmitter on exertion. That's why there is a weakness on exertion. There will be weakness on exertion. Now when the patient will take a rest, so when the patient will take a rest, what will happen? The release rate will be somewhat decreased, and the rate of synthesis will be increased. So when the rate of synthesis will be increased, then what will happen? There will be more release of neurotransmitters. There will be more release of transmitters subsequently. So the patient will improve on rest. So two important characteristics of a myasthenia gravis, that there will be muscular weakness. Second thing is that the muscular weakness will become worsen on exertion. Third thing, that muscular weakness will improve on taking rest. Got it? Now, what are the changes which is happening at the neuromuscular junction? If you see a patient of myasthenia gravis, so you will see that there will reduce number of acetylcholine receptors at the neuromuscular junction. There is a loss of postsynaptic fold. You can see here the loss of postsynaptic fold, and there is increase in this synaptic space. Now the synaptic space will be more than 20 to 30 nanometer wide. Diagnosis of this myasthenia gravis is made by a test which we call them as repetitive nerve stimulation test. So if you here what you do, you are stimulating a nerve and then you are seeing the muscular contraction there. So you will see in a repetitive nerve stimulation test that initially the amplitude of this action potential will be more, but when you repeatedly stimulate the nerve, then the amplitude of action potential in the muscular membrane, that will subsequently decrease. Got it? Reason for this decrease is that there less and less neurotransmitter would be released subsequently. As a result, there will be less and less depolarization of the muscle membrane. As a result, the amplitude of the action potential which is generated at the muscle membrane, that will subsequently decrease. Now, treatment for this myasthenia gravis is that the two acetylcholine esterase inhibitors, physostigmine and neostigmine. So here, what they do, these drugs, they bind with the acetylcholine esterase and they inhibit it. And because of this inhibition of acetylcholine esterase, what is happening? The destruction of acetylcholine is somewhat prevented. And since the destruction of acetylcholine is somewhat prevented, that means acetylcholine can act over the receptor for longer time. And because of this longer time of the acetylcholine, they will act over the membrane and they will cause depolarization of the muscle, and because of that there will be muscular contraction. So here the survival or the rate of destruction of acetylcholine is somewhat slow by inhibition of this enzyme, acetylcholine esterase. An example of this acetylcholine esterase inhibitor are neostigmine and physostigmine. So they are the drug. Similarly, there are some acetylcholine esterase inhibitors which are used in war also as nerve gas. For example, I tell you diisopropyl phosphate. So here this diisopropyl, what they do, they act over this acetylcholine esterase and they inhibit it for longer time. And because of inhibition of these acetylcholine esterase for longer time, for V, there will be continuous depolarization of the muscle, and because of this continuous depolarization of the muscle, there will be sustained contraction and there is the tetany. And here the most of the patient are dying because of laryngospasm. Because of sustained contraction, the muscle of the larynx will contract, and when the muscle of the larynx will contract, then what will happen? The patient is not able to breathe. So some of the this acetylcholine esterase inhibitor, they are used in war also as nerve gas. Second disease, we call them as Lambert-Eaton syndrome. It is also an autoimmune disease, and here the antibody is generated against the calcium channel which is present at the nerve terminal. So here the patient, what will happen, since the antibodies are acting over the calcium channel, so what is the problem is there? The amount of calcium which is required to be increased at the nerve terminal so that neurotransmitter could be released, that increase is not happening. As a result, the amount of neurotransmitter release will be less. And when the amount of neurotransmitter release is less, there will be less depolarization. As a result, there will be muscular weakness. So here the patient will complain that there is a muscular weakness, but here the patient will say that on exertion, this muscular weakness improves. The reason is that you see what is happening. Some antibodies are there, and these antibodies are acting over the calcium channel. As a result, there's a less amount of free calcium channel is available. So less increase in calcium is happening at the axon terminal. So less release of neurotransmitter is happening. So this is the reason for weakness. Now, when you are stimulating or when you are doing repetitive activities, then what is happening? There's a repetitive nerve stimulation. So when there is a repetitive nerve stimulation, that means the large amount of action potential is reaching at the nerve terminal. And since when the large amount of action potential is reaching at the nerve terminal, this calcium channel, whatever the left out calcium channel is left there, they will get activated for more and more time. As a result, there will be more and more accumulation of calcium at the nerve terminal. And when there is more and more accumulation of calcium channel at the nerve terminal, so there will be more and more release of calcium, more and more release of the neurotransmitter. So this weakness will improve on exertion. So this is the difference between the two diseases, myasthenia gravis and Lambert-Eaton syndrome. In myasthenia gravis, the weakness will increase on exertion, and in Lambert-Eaton syndrome, the weakness will improve with exertion. And just like myasthenia gravis, the diagnosis of this test is also based on repetitive nerve stimulation test. So on repetitive nerve stimulation test, you will see initially here the amplitude of the action potential of the membrane potential that will increase when there is a repetitive nerve stimulation. In myasthenia gravis, the amplitude was decreasing on repetitive nerve stimulation. But here the amplitude will be increasing on repetitive nerve stimulation. Now we will discuss about few agents which act over the neuromuscular junctions. So first is your botulinum toxin. So botulinum toxin, this is being released by a bacteria which we call them as Clostridium botulinum, and this toxicity happened because of food poisoning. So this bacteria Clostridium botulinum, this releases this toxin botulinum, and this toxin will act over presynaptically, and because of action of this toxin and diffusion of acetylcholine vesicles with the presynaptic membrane is blocked. And since this acetylcholine vesicles, they are not fusing with the presynaptic membrane, so there will be no release of neurotransmitter. So this is a kind of neurotoxin, botulinum toxin. Then there is another toxin which we know as conotoxin. Conotoxin act presynaptically, and where they act, they block the voltage gated calcium channel. So when there is a blockage of voltage gated calcium channel, so there will be no increase in the calcium concentration inside the nerve terminal, and when there is no release of calcium ion channel in the nerve terminal, so there will be no release of neurotransmitter. Another is your puriform drugs, and one very important example of this puriform drug is your D-tubocurarine, and this drug acts postsynaptically, and they act just like acetylcholine receptors. They act over the nicotinic receptor, and it is a type of competitive inhibition, meaning that these drugs, this tubocurarine, they compete with acetylcholine to bind over the nicotinic receptor. So they come and they bind over the nicotinic receptor. So there is less amount of you can say there's a less amount of receptor left for acetylcholine to bond. So what will happen? There will be muscular relaxation. So this D-tubocurarine is used as muscle relaxant. Got it? They come in the category of non-depolarizing muscular relaxant. So they are used in clinical practices when you are doing some kind of surgeries. So there you need muscle to get relaxed. Got it? For example, when you are doing some procedure, for example, when you are putting a tube in the larynx, so you need that the muscle of the larynx should be relaxed so that the tube can easily go inside the larynx. So, or you are doing some kind of stitching in some area where there's a loss of where the muscle has contracted, or there's a muscular spasm. So you need that muscle spasm to go, and muscle should relax so that the stitching process becomes easier. So there you can use this kind of drugs. So where they will compete with the acetylcholine, and there will be muscular relaxation. Then another type of drug which is commonly used in anesthesia, your succinylcholine. So this succinylcholine, they come in this is a type of muscle relaxant, and they come in the class of depolarizing muscular relaxant. So here they act postsynaptically, and here they act just similar to acetylcholine. These acetylcholine, they bind with the nicotinic acetylcholine receptor, and after binding, there is change in the configuration of nicotinic acetylcholine receptor, and because of this change in configuration, the sodium channel will open, and there is an entry of a large amount of sodium. But the difference is that these succinylcholine are not be destroyed by the acetylcholine esterase, which is present at the synaptic space. And since they are not destroyed by acetylcholine esterase, they keep on acting. They keep the sodium channel open for a very long time. Now, what will happen when the sodium will get? We know that the sodium ion channels, they open because of depolarization, but they also get closed because of depolarization. So first, because of opening of sodium channels, there will be depolarization of the membrane of the muscle. But because of the persistent depolarization, what will happen? This sodium channel will get inactivated. And when the sodium channel gets inactivated, there will be no further depolarization. Got it? And because of that, when there is no further depolarization, that what will happen? There will be relaxation. So in this type of drug, there is the initial twitching of the muscle because of depolarization. But because this type of drug causes persistent depolarization, so there will be paralysis. Whatever. So they act like a acetylcholine, but the difference is that they are not destroyed by acetylcholine stress, and since they are not destroyed by acetylcholine stress, they will cause persistent depolarization, and because of this persistent depolarization, there will be relaxation of the muscle. So initially, this drug will cause a slight contraction, which appear as twitch, and then there will be muscular relaxation. Bangaru toxin is a neurotoxin which is present in snake. It's a type of venom, snake venom, and they act over the postsynaptically over the acetylcholine receptor. Here, they bind with the acetylcholine receptor, just like that, and they will cause muscular relaxation, complete muscular relaxation. Got it? So they will bind. And this binding of Bangaru toxin is, you can say, it is they bind in a irreversible manner. They are not removed by acetylcholine stress, so they will cause persistent depolarization, and because of this persistent depolarization, there will be relaxation of the muscle. So these are the some agents which are acting over your neuromuscular junctions. Okay. So today we have discussed about neuromuscular junction. So there we have seen what the neuromuscular junction is, about the physiological anatomy of the neuromuscular junction, then about the events which is happening at the level of neuromuscular junction, then a few things about the acetylcholine which is released at the nerve terminal, then few things about the receptor of acetylcholine over which is present at the neuromuscular junction, and that is your nicotinic acetylcholine receptor there. And then we have seen two important diseases, myasthenia gravis and Lambert-Eaton syndrome. What is the difference between them, these two diseases? What are the causes of these two diseases? And then about few agents which act over neuromuscular junction. Okay. So with this, we complete about the neuromuscular junction. Now coming to today's topic of discussion, that is your about the muscle. So we have completed about nerve. We have completed neuromuscular junction. Now coming to the muscle physiology. So we know that if you see the muscle, so whatever the muscle that is present in your body, they could be classified into three important groups. One is your skeletal muscle, second is your cardiac muscle, and third is your smooth muscle. Got it? So we will see that the skeletal muscle is forming about 40% of your body weight, and cardiac and smooth muscle, they are constituting about 10% of your body weight. The skeletal muscle is present throughout your in the skeletal structure. All your things, they are voluntary in nature. That means whether the muscle will contract or not. If you contract, it will depend upon your own weight. If you want to pump your bicep, you can pump. If you want to relax your bicep, you can relax. That is up to your will. Then cardiac muscle, they are of course present in your heart. All your atria, ventricle, they are composed of your cardiac muscle. The smooth muscle, rest of the muscle are your smooth muscle. All the muscle which is present in your airway, in your GI tract, in your urinary tract, or the muscle which are present in your reproductive tract, your uterus and everything. They are just they are your smooth muscles. Now, before discussing about the skeletal muscle, let's see what are the functions of skeletal muscle. So I know that most of the functions you will be knowing from your +2 knowledge. So skeletal muscle, they produces your skeletal movements, meaning that the movement of your hand, movement of your leg, or movement of your back, movement of your head. It is because of contraction of some muscle, and because of this contraction, there is a flexion or extension of any of the joints that lead to the movement. Second important function is your maintenance of your body position and posture. The maintenance of your body posture and position is because of contraction of your active gravity muscles, which is present in your lower limb and your upper limb, and the muscles of your back. So they are the muscles which are making you stand against your gravity. If these muscles are not contracting, then you will not be able to stand against the force of gravity and you will fall down. Then they, of course, they support your soft tissues. If you see there in your skeleton, if you see the muscles of your abdomen cavities or the muscle of your thorax, so here you will be seeing that some skeletal muscles are there, and they are protecting all the viscera which is present in your thorax, whether it's your heart or lungs, or the viscera which is present in your abdomen, your GI tracts, your kidney, or your stomach, your pancreas, your liver. They all are somewhat protected and supported by this. Then maintenance of your body temperature. Muscles have very important function with respect to maintenance of your body temperature. One of the very important things that they see, whenever the muscle will contract, they will generate heat, and this heat is used to raise your body temperature. So one of the very important things that you will observe that whenever you come to your class, whenever you come out in your winter time, you will see when the temperature is very low. People start shivering, है ना? So this shivering is a physiological thing. So whenever this shivering is manifestation of your muscular contraction and relaxation. So whenever you shiver, you move your limbs, and there is the movement of your muscle. And because of this contraction of your muscle, there is a generation of heat that help you to fight the cold weather of your outside. And last important function that they act as a storehouse of the nutrients. So you will see that nutrient like glycogen. So large amount of glycogen are stored in your muscle. Apart from that, some fatty acids, fats are also stored in muscle. So these glycogen or fat, they are used in case of emergency, so, or whenever you're fasting or something. So whenever the glucose level will decrease, the glycogen which is stored in your muscle or fat which is stored in your muscle, they will break down into a smaller unit, that is your glucose, which will provide you energy. Now, if you see a muscle belly, suppose you take a muscle belly, suppose you take your biceps. So what are the structure which is present in the muscle belly? You will see, of course, there may this muscle belly is made up of muscle muscle fibers, and then these muscle fibers will be surrounded by some connective tissues will be there. Then there will be some nerves, and then there will be blood vessels, which is present in the connective tissues, which is surrounding your muscle fibers. And it is through these blood vessels the blood supply to the muscle is ensured, and through this blood supply the oxygen or the nutrients which is reaching the muscle. So this is the composition of a muscle belly. That muscle belly is composed of muscle fiber, connective tissue, nerves, and the blood vessels. Now, if you see a muscle, then you will see that the muscle fibers are there, and these muscle fibers are somewhat surrounded by connective tissue. Just like a neuron, we have seen that there the nerve is surrounded by a connective tissue, which we used to call as endomysium. In the muscle, these connective tissue we call them as endomysium. And then many of these muscle fibers, they come together, they form a bundle, and this bundle is again surrounded by connective tissue, which we call them as perimysium. In the nerve, used to call them as perineurium. Here we call them as perimysium. Now many of the muscle fascicles, they come together, got it, and these bundles of the muscle fiber, they are covered by another connective tissue, which we call them as epimysium. So three connective tissues are there which is surrounding the muscle. An individual muscle fiber is surrounded by connective tissue which we call them as endomysium. A bundle of muscle fiber are surrounded by a connective tissue which we call them as perimysium. And then many bundles, they come together, and then they are surrounded by connective tissue which we call them as epimysium. Now if you see this functional organization of muscle fiber, so here you can see this is a muscle belly, got it, and these are individual muscle fibers are there, got it. Now if you see a individual muscle fiber here, they are covered by a connective tissue which we call them as endomysium. Then many of these muscle fiber, they come together and they form a bundle, and this is covered by a muscle, the connective tissue, which we call them as perimysium. Then many of the bundles, they come together here and they are covered by a connective tissue. We call them epimysium. So if you see the functional organization of a muscle, then here in the middle, first you have muscle fiber, then they are being arranged in bundle form, which we call them muscle fascicles, and then many muscle fascicles, they come together and they form muscle bed. Now, if you see an individual muscle fiber, so this individual muscle fiber is covered by a plasma membrane, which we call them as sarcolemma. Okay, it is just like a plasma membrane of any individual cell, but they are in muscle, they be named with a different name, which we call them as sarcolemma. And inside of the sarcolemma, they have sarcoplasm. It is just similar to cytoplasm of a cell. So in an individual muscle fiber is covered by a membrane, which we call them as sarcolemma. Inside you have sarcoplasm, and in this sarcoplasm you have all the organelles which any cell is having. That means they are having mitochondria, Golgi apparatus, endoplasmic reticulum, and inside the sarcoplasm they have many longitudinal filaments are there, and these are, we call them as myofibrils. Got it? So inside of the muscle fiber you have you have myofibrils, and these myofibrils, they are made up of structure which we call them as myofilaments, and these myofilaments are basically the contractile unit. Whatever the muscular contraction is happening, they take at the level of myofilaments. Because of the contraction of the myofilaments, the myofibril will contract, and because of the contraction of many myofibrils, the muscle fiber will contract, and because of contraction of the muscle fiber, your muscle belly will contract. So in the muscle fiber, inside of the muscle fiber, you have sarcoplasm, and in the sarcoplasm you have large amount of myofibrils. Okay, you have myofibrils are there, and these myofibrils are made up of myofilaments. Okay, and these myofilaments are contractile units, and it is these myofilaments which are contracting and relaxing, and in response of this contraction and relaxation, your muscle fibers are contracting. So here you can see again in this diagram here how the muscle fiber are being organized. This is the individual muscle fiber, and this individual muscle fiber is arranged in a bundle, a fascicle, and it is covered by perimysium. And this many of these bundles, they come together and they form a muscle belly, which is covered with surrounded by a connective tissue, which is called by epimysium. Got it? Now, if you see the muscles, they are attached to your bones. That means your skeleton of your body. Got it? So most of the this muscle, they are attached to the bones through a filamentous structure, which we call them as tendons. So the muscles are the origin on exertion or insertion of the muscle to the bones is because of these tendons. And the muscle, and if you see the muscle fiber between these two tendons which are attaching to the bone, all these muscle fibers are parallel to each other. And this parallel arrangement of the muscle fiber help us to add the force of contraction of each of the individual muscle fibers. So when the force of contraction of each of the individual muscle fiber, they add up, they produce large amount of force, and because of that there could be movements. Okay. Now coming about the embryology of muscle, how the muscle is developing. So you will see the skeletal muscle, the skeletal muscle fibers, they develop because of the fusion of mesodermal cells, which we call them as myoblasts. So there are many these mesodermal cells, which we call them as myoblasts. Many of these myoblasts, they come and they fuse, and because of fusion of this mesodermal cells, there's a formation of this skeletal muscle fibers. And since there is a fusion of a large amount of mesodermal cells, which we call them myoblasts, that's the reason why if you see a individual muscle fiber, they're multinucleated, because they are formed because of fusion of mesoblast cells. So many mesoblast cells, they fuse with each other. Each mesoblast cell are having a nucleus. So whenever the large amount of neuroblasts cells they are fusing to form an individual muscle fiber. So that's the reason why the individual muscle fibers are multinucleated. So if someone asks you why the muscle fibers are multinucleated, you say because they develop because of the fusion of large amount of mesoblast cells. That's why they are multinucleated. So before discussing about the further discussion about the individual muscle fiber, some of the basic character you must be knowing, and that is if you see an individual muscle fiber, they are long, thin, cylindrical in shape, and they are multinucleated. We know that why it's multinucleated. They are striated. Striated means that if you see the muscle fiber, individual muscle fiber under a microscope, so you will see that there's a pattern of dark and light band. So this gives a striated appearance. Dark band, light band, dark band, light band. So because of that, they have a striated appearance. There's no syncytial bridge between the cells, meaning that there's no branching. That means it's not like that one muscle fiber is giving a branch and this branch is going to make contact with another muscle fiber. So this is not happening at the level of the skeletal muscle. This syncytial bridge is there in your cardiac muscles, or you can see we will see later on in the smooth muscle. And then the next property that they are voluntary. Of course, they are innervated by your motor neurons, which are coming from your ventral root of spinal cord, and they are under your conscious control. So whenever you want to contract a muscle, you can contract. Whenever you want to relax, you can relax. So that is the contraction and relaxation is under your control. This is a big difference between your cardiac and smooth muscle. Your heart is beating. It will beat. It will not stop when you will want, and the rate of beating will not increase when you wish. No. Similarly, the GI movements, the GI, your gastrointestinal tracts are made up of smooth muscle. They have their own intrinsic rhythm of contraction and relaxation, and that can't be changed by your voluntary reason. And the rate, as compared to this cardiac and smooth muscle, they are showing a rapid contraction. So the contraction and relaxation of cardiac muscle of your skeletal muscle is more rapid as compared to cardiac and smooth muscle. As we will go further in this lecture, then we will slowly we will know the differences between skeleton, cardiac, and smooth muscle. So the basic properties of the skeletal muscle: they're long, thin, cylindrical in shape. They're multinucleated. They're striated in appearance. There's no syncytial bridge between two individual muscle fiber. They are voluntary in nature, and they are showing rapid contraction. Now coming to the structural detail about this individual muscle fiber, as you told before, the cell membrane of muscle fiber, we call them as sarcolemma. This sarcolemma is just like any plasma membrane. Okay, so they are made up of plasma membrane, and apart from that, there is a thin layer of polysaccharide material, which is having large number of collagen fibrils that is present over the surface of these plasma membrane. So, okay. So if you see the plasma membrane of muscle, this is known as sarcolemma. They have a lipid bilayer with protein embedded. Along with that, on the outer layer, you have a thick layer of polysaccharide, which is rich in collagen fibrils. So here you can see an individual muscle fiber, and in the individual muscle fiber, the outer covering is known as sarcolemma. Inside the individual muscle fiber, you can see large amount of longitudinal structures are there, and these longitudinal structures are, we call them as myofibrils. Now, most of the if you see the structural component of a skeletal muscle, they are similar as compared to any individual cell. That means they are having sarcolemma inside. They have sarcoplasm, which is similar to cytoplasm. They have nucleus. They are having all the mitochondria, Golgi apparatus, endoplasmic reticulums are there, which are surrounding these myofibrils. Apart from that, they have another important characteristic, that they have T-tube or transverse tissue. Now, first you know that what is the before knowing what actually T-tubules are, you should know what is the special requirement of a skeletal muscle. For contraction of skeletal muscle, there is some special requirement. Special requirement is that you see we have seen that in the neuromuscular junction, the electrical impulses is being transmitted from your nerve terminal to the membrane of muscle. and at the level of membrane of the muscle there is a depolarization. And this once this action potential is created, this is traveling throughout the sarcolemma of the muscle fiber. Now for contraction of a muscle fiber, this depolarization must penetrate the sarcolemma, and they should go in the neighborhood of myofibril, because see, these myofibrils are having the myofilaments which are contractile element. So if this depolarization is not reaching to the myofibril, your myofibrils will not contract, and when it is not contracting, then how the individual muscle fibers will contract. The problem is that these myofibrils are present inside the muscle fiber, and this depolarization is running on the surface of muscle fiber. That means there must be some anatomical changes because of which this depolarization must penetrate to the inside of muscle fiber, and this depolarization must reach to the myofibril. And for this, the nature has devised a change in the sarcolemma of muscle fiber, and this change we call them as transverse tubule or T tubule. Here what is happening, the sarcolemma will invaginate to the inside of muscle fiber, and they will invaginate and they penetrate to the other side of muscle fiber. So this T tubule or transverse tubule, they are invagination of muscle fiber. So here you can see this is sarcolemma, and here you can see this sarcolemma has invaginated, and they will move, they penetrate throughout the muscle fiber to the opposite end. Got it? So this transverse tubule or T tubule is invagination of sarcolemma, and the purpose of this invagination is that we show that the wave of depolarization should penetrate inside the muscle fiber so that it reaches the myofibril and the myofilaments. And this T tubule is just the extension of extracellular space. Here you can see this T tubule is in continuity with the fluid which is present on the outside of the muscle fiber. So they are in contact with the extracellular fluids. And the function, we have know that the function of this transverse tubule is that they must carry this depolarization to the inside of muscle fiber in the neighborhood of myofibril, in the neighborhood of the myofilament, so that this contractile elements they will get depolarized and they will contract, so that the muscle fiber will also get contracted. Now, coming to another concept, which we call them as sarcotubular system. Now, to understand the sarcotubular system, two things are important for muscle to contract. One thing is that there should be depolarization at the level of myofilaments, and second thing is that in response to this depolarization, there must be increase in the concentration of calcium inside the muscle fiber. When these two things happen, that means the depolarization is reaching at the level of myofilaments, and in response to this depolarization, when the amount of calcium channel is increased about 10,000 times, then there will be contraction. We will see later on what is the importance of calcium when we will read about excitation contraction coupling. At this level, just keep in mind that for a muscular contraction, two things are required. First thing, that there should be depolarization, and this depolarization should reach at the level of myofibril. Second thing is that in response to this depolarization, there must be release of calcium inside the sarcoplasm of muscle. Then only the muscle can contract. Okay. How this calcium will lead to contraction, that is a different topic that we will discuss upon in excitation contraction coupling. I am telling you this just to make you understand what is sarcotubular system. So sarcotubular system is a specialized system for internal conduction of depolarization within the muscle fiber and for calcium release. So this sarcotubular system does two things. One thing, they carry the depolarization to the inside of muscle. Second thing, they will cause release of calcium in the sarcoplasm. And this sarcotubular system is made up of two things. One thing is your T tubules and another is your sarcoplasmic reticulum. Sarcoplasmic T tubules we have discussed, that is your transverse tubule that will cause internal conduction of depolarization. Okay. Second purpose of sarcotubular system was to release the calcium. And in this release of calcium is because of sarcoplasmic reticulum. Sarcoplasmic reticulum are just smooth endoplasmic reticulum. In any cell, we have smooth endoplasmic reticulum. These smooth endoplasmic reticulum, which is present in the skeletal muscle, we call them as sarcoplasmic reticulum. So here you can see in this structure that this sarcoplasmic reticulum, this blue type of color you can see, this is a muscle fiber, and this muscle fiber are surrounded by network of structure, and this network of structure which is blue in color, we call them as sarcoplasmic reticulum. So sarcoplasmic reticulum are smooth endoplasmic reticulum. They run longitudinally along the surface of myofibril and they surround the whole of the surface area of myofibril. And this sarcoplasmic reticulum is composed of two components. One is your terminal cisternae and another is longitudinal tube. Terminal cisternae are basically their large dilated sacs or chambers which lie in close proximity with T tubules. So here you can see here in this diagram, this is your T tubule, and in the adjacent to this T tubule there is large dilated sacs of these your sarcoplasmic reticulum. So these large dilated sacs which are lying adjacent to your T tubules, they are what we call as terminal cisternae. Got it? Non-longitudinal tubule, they run along the surface, they run longitudinally along the surface of myofibril and they surround the myofibrils. So these are known as long longitudinal tubule. Here you can see here the just in the neighboring of terminal cisternae, these long longitudinal tubules are there which are covering the surface of myofibrils. Got it? So they are present. Now these sarcoplasmic reticulums, what is their function? They function as a storehouse of calcium. Got it? So they are having large amount of calcium inside, and whenever there is a depolarization at the level of myofibrils, so in response to this depolarization, there is a release of calcium channels from these storehouse, from these sarcoplasmic reticulum. As a result, the concentration of calcium ion inside the sarcoplasmic reticulum, they are increased by about 10,000 times. And when this increase in concentration of calcium ions happens, then there will be contraction of the muscle. Okay. So we will discuss about that in excitation contraction technique. Today we are just discussing about the structural aspects of a muscle. Okay. Now another term which we call them as triad. Triad, what it's triad? It is a T tubule and the two adjacent terminal cisternae. So that a T tubule with two adjacent terminal cisternae, they form the triad. Got it? So this is white structure is your T tubule, and adjacent to this T tubule, this term dilated sacs or chambers are there, which we call them as terminal cisternae. So that all together they form a structure which we call them as triad. Triad means three structure. थ्री क्या है? एक बीच में टी ट्यूब्यूल है और उसके अगल बगल दो डायलेटेड सैक्स है, ठीक है, जिसको कहते हैं टर्मिनल सिस्टर्नाई. तो ये सब मिला के एक स्ट्रक्चर फॉर्म करता है व्हिच इज वी कॉल देम एज ट्रायड. We will see what triad do later on. Now coming to the next structure, that is your myofibril. So we have seen that muscle fiber is there, and inside the muscle fiber you have large number of longitudinally arranged structure which we call them as myofibrils. So this myofibrils, they are composed of further smaller subunits, and these smaller subunits we call them as myofilaments. Okay, and these myofilaments are basically the contractile unit of a skeletal muscle. So in response to depolarization, there is contraction at the level of myofilaments, and because of this contraction, the myofibril will contract, and when myofibril will contract, the muscle fiber will contract. Now these myofilaments are of two types. One is your thick filament, another is your thin filament. Got it? So in one myofibril, it is said that about 3,000 thin filaments are there and about 1,500 thick filaments there. These thin filaments are made up of protein, predominantly made up of protein, which we call them as actin protein. And thin filaments are predominantly made up of protein, which we call them as myosin protein. Got it? So myofibrils are there inside the muscle fiber. Myofibrils are made of contractile unit, which we call them as myofilaments. Myofilaments are of two types, thick and thin filaments. Thick filament is made up of myosin, and thin filament is made up of actin. Now this thick and thin filament you will see. Here you can see your thick and thick filaments. So you will see that thick and thick filaments are arranged in a particular pattern there in the myofibrils. And you will see that for some up to extent there is a thin filament, you can say there is a interdigitation of thick and thin filament. So up to some area the thin filament will interdigitate or insulinate in the thick filament. So or you can say there's overlap, there's the interdigitation of thin filament in the spaces between the thick filament. Got it? So in some particular manner, the thick and thin filaments are arranged. We will see just now how they are arranged. Now coming to the muscle striation. So I told you before that when you see the skeletal muscle under microscope, then you will see that there is a pattern of dark band and light band, dark band and light band. So there is a striated appearance. So because of that, we call that the skeletal muscle is striated, muscular striation. And it is said that the light band is also known as I band or isotropic band. Dark band also known as anisotropic band or A band. Got it? Now, why the light band is called known as I band or isotropic band, or why the dark band is known as anisotropic band or A band? To understand this, you see, this is your dark band, and this area which is having a thick filament and thin filament. So this is your dark band, and this area which is made up of only thin filament, this is your light band. Now what happened actually, the isotropic means, the word meaning of isotropic is that it has uniform property. So if you see the light band, they are made up of only thin filaments. So there is the property of this area is uniform. And if you do the phase microscopy of this area, then the structure which is having uniform physical property, they show single refraction. And because of this single refraction, they appear light in color. But if you see the dark band or A band or anisotropic band, this band is made up of both thick filament and thin filament. That means the physical property of this area is not uniform. They are made up of two filaments, thick filament and thin filament. And because of the interdigitation of this thick and thin filament, this A band or dark band doesn't have a uniform physical property. And when a structure which is not having a uniform physical property, if you do phase microscopy there, then there will be a double refraction, and because of that double refraction, they appear dark in color. So while the light band is known as isotropic or I band, we call them isotropic because they have a uniform physical property. They are made up of only thin filament. And while the dark band, we call them as anisotropic or A band. Why? The reason is that they don't have a uniform physical property. This band is formed by interdigitation of thin filament and thick filament. So they don't have a uniform physical property. That's why they are called as anisotropic band or A band, and they appear dark in color on phase microscopy. And light band is known as isotropic band or I band, and they appear light in color. So now here you can see this is the thing that appear in your microscope. So this is your dark band and this is your light band. Got it? And in the dark band you will see there's a small area of light band, and this small area of light band we call them as H band. Got it? We will see why there is a light band. And if you see in the light band, or if you see in I band, so you see in the I band, there is a dark line is there. Got it? Here you can see this is I band, and in this I band there is dark line. This dark line is known as Z line. So this is the dark band. Adjacent to that, you have light band, and this light band is divided into two by this Z line. Okay, and in the dark band, you have in the central part of your dark band or A band, you have a light band which we call them as H band. Okay. So now we will see about dark band and isotropic band. Dark band, anisotropic band. Reason is that the anisotropic, why? Because this area has a non-uniform physical property, because it is made because of interdigitation of thick filament and thin filament. If you see the light band, if you see the length of this band, it is 1.5 micrometer, and the lighter area in the center of dark band is known as H band. Okay. So why there is a light area? You see that the uh, this thin filament is interdigitating into this thick filament, but they are not reaching in the center. There is a small space where in the light band they are made up of only thick filament. In this space you don't have thin filament. Got it? So this area where you have only thick filament, so that means this area has the central area of your dark band has a uniform property, and they have only thick filament. So this area appear as light area. Got it? And in the center of this H band, you have a line which we call them as M line. Got it? And pseudo X zone means that the M line plus a narrow light area on the either side of this M line, which we call them as pseudo X zone. They are just terminology. Important thing for you to remember that there is a dark band and light band. Why dark band is dark? Why light band is light? You should know. What anisotropic mean? What isotropic mean? In the center of your dark band, there is a light band, which we call them as H band. Why the H band is light? You should know. Okay. Now, light band, if you see, the length of light band is 1 micrometer. It is about 1 micrometer. And in the center of the light band, you have a dark line, which we call them as Z line. Now, it is this Z line to which thin filament are being anchored. What is this Z line is made up of filamentous proteins, and it is this to this filamentous protein the actin filament or thin filaments are anchored. Got it? So one light band is divided into two by Z line. Got it? This is your dark line. This is dark band. This is light band, and surrounding on the other side also your light band. And then this light band is divided by Z line here, and then this Z line is divided. This light band is again divided by Z line there. Now coming to the concept of sarcomere. Sarcomere is basically the portion of myofibril that lies between two successive Z lines. Got it? This portion of myofibril we call them as sarcomere. And this sarcomere is the structural and functional unit of muscle, meaning that the contraction is happening at the level of sarcomere, and there are many sarcomere which are longitudinally arranged along the myofibrils, and many sarcomere they are contracting, and because of this contraction the myofibrils are contracting. So the portion of myofibril between two successive Z lines we call them as sarcomere. So in this diagram you can see, so this is your dark band, this is Z line here, and this is Z line here. So this portion of myofibril between this Z line and this Z line is called as sarcomere. And this is the structural and functional unit of myofibril, just like neuron, which is structural and functional unit of nervous system. So if you see this sarcomere is made up of one Z line, and half of one A band and half of the I band on the adjacent sides. Got it? So one sarcomere is made up of one A band and half of two adjacent I bands. Got it? So sarcomere is made up of one complete A band and two half of I band, which is adjacent to it. So here you can see this is structure of sarcomere. So this is your A band, okay? Then half of uh, I band on this side and half of I band on this side. Now this A band, the length is about 1.5 micrometer, and the length of I band was complete I band was 1 micrometer. But here you have half, that means 1.5 micrometer here, 0.5 micrometer here, and 0.5 micrometer on this side. So if you see the total length of a sarcomere, it is 1.5 plus 0.5 on this side, 0.5 on this side. So in a resting state, the length of sarcomere would be 2.5 micrometer in here. Got it? And in skeletal muscle, here at this point, remember that triad is located at the junction of A and I band. This is junction of A and I band. This is A band and this is I band. At this junction, this transverse tubule is penetrating. So this triad is locating at the junction of A and I band. Got it? Okay, this is your A and I band. That's why a skeletal muscle is a single sarcomere is having two triads in skeletal muscle. Now coming to the characteristics of your thick and thick filament. Now we have seen that myofibrils are there, and myofibrils are made up of myofilaments, and these myofilaments are again classified into thick filament and thin filament. Now, thick filaments are basically made up of myosin molecules. So if you see the thick filament, it is if you see the diameter, it is 10 nanometer in diameter, and one thick filament is composed of about 500 or more of individual myosin molecules. Now, if you see myosin molecules, so this one myosin molecule is composed of 6 polypeptide chains, and in this 6 polypeptide chains are 2 are heavy chain and 4 are light chain. So if you see, this is a typical structure of a myosin molecule, and if in a typical myosin molecule, they have one tail and two heads. So this is the tail and this is two heads of myosin. Okay. So here the tail is made of a heavy chain. So these two heavy chain they are arranged in double helical form, just like DNA, and they form the tail. So two heavy chain arranged in a double helical form, they are making this chain. Now a head is made up of a one heavy chain and two light chain. So what is happening at one end, the head is folded bilaterally. They have one of the heavy chain is folded bilaterally to form a globular structure, and to this globular structure, two light chain are arranged. Okay, so this is forming one head, and then you have another head. It is again formed by the folding of a heavy chain and two light chain. So remember, a myosin molecule is having is made up of 6 polypeptide chain. Two are heavy chain and two are light. Four are light chain. Uh, if you see the structure, they have one tail and two head. Tail is made up of heavy chain, and these heavy chain they are arranged in a double helical form. Head is made up of one heavy chain and two light chain. Got it? Now, what is the function of myosin head? Myosin head is very important with respect to contraction. One important function that they act as a ATPase enzyme. ATPase enzyme means that they will break the ATP molecule. So whenever there is muscular contraction, there is a requirement of energy. So the ATP molecule will bind to this ATPase head, and this ATP molecule will be disintegrated, will be broken down into ADP and phosphate, and there is a release of energy. Plus, it is this myosin head which is making attachment with the actin filaments, a thin filament. And when the head of myosin get attached with actin filament, there is a contraction of muscle. We will see that when we discuss about excitation contraction coupling. At this time, just remember the myosin head has two functions. One is that they break down the ATP into adenosine diphosphate, a plus phosphate, and cause release of energy. Second thing, they are the one which is making contact with actin filament, and after making contact with actin filament, the myosin head moves, and there is a movement of actin filament over the myosin filament, and because of that, there is a muscular contraction. Now coming to thin filament, of course, the diameter is less than thick filament. It is about 5 nanometer, and they are composed of three important proteins: actin, tropomyosin, and troponin. Okay. Now, actin filaments, we have seen in sarcomere, we have seen that actin filament, they are attached with Z line. So they are, and from the Z line, they protrude in both the direction, and they are placed in the space between your thick filament. So here you can see the thick filament is there, and then there is this thin filament. This thin filament is made up of actin, and this actin is attached to Z line, and from the Z line they are protrude inside in the space between the thick filament, that is your myosin filaments. Now, if you see the actin filament, they are made up of two long polypeptide changes, and these two long polypeptide changes are known as F-actin molecule. F-actin molecule, we call them as fibrous actin molecules. Got it? So these two polypeptide chain, individually, which are called as F-actin molecule or fibrous actin molecule, they are arranged in just like a double helical structure. So here you can see that these blue dotted lines, so they are F-actin molecules which are arranged in double helical form to form a actin filaments. Now, if you see one strand of actin, you will see that they are made up of a smaller globular proteins, and these smaller globular proteins, you can see they are getting polymerized and they're forming one strand, and two of these kind of strands that together they make actin filament. Now these the globular proteins which are polymerizing and making F-actin molecule, they are known as G-actin molecule, okay, or globular actin molecule. So many globular actin molecules, they polymerize together and they form F-actin molecule or fibrous actin molecule. Now two of these F-actin molecule or fibrous actin molecule, they arrange themselves in a double helical form to form an actin filament. Now you will see in this G-actin molecule, there is uh, a molecule is attached which we call them as adenosine diphosphate molecule. So each of this G-actin molecule, one molecule of adenosine diphosphate is attached, and this adenosine diphosphate molecule, which is attached to the G-actin molecule, they act as the active site on actin filament. By active site, we mean that it is these site to which the myosin head is making attachment on the actin filament. Got it? So these sites on which the ADP molecule is attached, these are the active sites, and over these sites, the head of myosin is making attachment when the muscle is contracting. Now, if you take a cross section through A band, in the A band, that is your dark band, where you have both thick and thin filament. So you will see that each of the thick filament is surrounded by 6 of your thin filament in a regular hexagonal pattern. So this is the arrangement of the filaments which is there in myofibrils. So if you take a cross section along the A band, which is having both thick and thin filament, then for every thick filament there is 6 of the thin filament which is surrounding them in a hexagonal pattern. Now this second molecule which is present on the thin filament is your tropomyosin molecule. Now these tropomyosin molecules, you can see this purple type of filamentous molecule. These are purple type of filamentous molecule. You can see in thin filament. So these tropomyosin are located in the groove of F-actin helix. So you know that the F-actin molecule are arranged in a double helical form. whenever any two polypeptide chains are arranged in double helical form, there will be groove. So in these groove, long filamentous molecule is present, and these filamentous molecule, the filamentous molecule which is present in these grooves in thin filament, we call them as topomycin molecule. The function of topomycin molecule is that they lie on the top of the active site of actin of thin filament. So they cover the active sites on the actin filament, and because of it is said that till the active site is covered by the topomycin, there will be no interaction between the head of mycin and the active site of actin. So basically they act as a barrier between head of mycin molecule and the active site of actin. And till it is covering the active site, there will be no interaction between the head of mycin and the active site of actin. Now the next molecule which is present in the thin filament is your toponin molecule. Now this toponin molecule is attached intermediately along the actin filaments, and this toponin molecule has three subunits: toponin I, toponin T, and toponin C. Now this toponin I subunit, it is attached with actin filament. Toponin T is attached with tropomycin molecule, and toponin C subunit, they attaches with calcium ion. Now if you see the toponin, you will see that one subunit of toponin is attaching with actin, and another subunit of toponin is attached with tropomycin. So basically this troponin acts as a glue between actin and tropomycin. So because of troponin, the tropomycin is attached with actin. Got it? So and this tropomycin is covering the active sites of the actin. Now the another subunit of troponin is your troponin C, which attaches with calcium. Now what happened actually whenever there is a muscular contraction, whenever there's a suppose there's an impulse coming through the nerve, and this impulse goes to the membrane of the muscle, and this impulse will create an action potential along the membrane of the muscle. Now this action potential will travel along the T tubules to the level of myofibrils, and in response to this depolarization, we suppose there is a release of calcium ion from the sarcoplasmic reticulum of skeletal muscle. Now this calcium will go and they will bind with troponin C, the subunit troponin C. And because of binding of this calcium with troponin C, what will happen? There will be some conformational changes. And because of this conformational changes, the tropomycin will move from its location, from its place. And when the tropomycin move from its place, what will happen? The active site of actin will be exposed. And when the active site of actin will be exposed, the head of mycin will attach with the active site of actin. And now the head of mycin will move. And when the head of mycin will move, the actin filament will also slide over the thick filament. And then there will be contraction. We will discuss in detail about that. Just I wanted to tell you at this level what is the importance of calcium. Here, I have told you before that for muscular contraction, two things is required. One thing is your depolarization, second thing is calcium concentration. So the calcium, the role of calcium is that they will come and they will bind with troponin C, and this will cause the conformational changes. And because of that conformational changes, the tropomycin will move from its location, and because of movement of that, the active site of actin will be exposed and the head of mycin will bind, and then there will be contraction. So a thick filament is made up of actin filament, tropomycin, and troponin. The function of tropomycin is to cover the active site of actin, and the function of troponin is to bind the tropomycin over troponin, and the function of calcium is to cause conformational changes in troponin, because of which the tropomycin can move from its location, and that can lead to exposure of active site of actin for the head of mouse. So apart from these, some individual proteins are also present in the muscle fiber. Uh, for example, you have one molecule which we call them as titin. We have discussed about actin, myosin, uh, then troponin, tropomycins. There are some other molecules are present also in the muscle. So one of them is titin. So this titin are protein strands which are composed of filamentous molecule. So here you can see, and this titin molecule they reaches from the tip of thick filament to the Z line. So here you can see this is titin. So this titin molecule is made up of filamentous molecule, and important function that is it attaches the end of thick filament to the Z line. So you can see this titin is attaching the end of thick filament to the Z line. The function of this titin molecule is that the side by side the relationship between thick and thick filament is because of this titin molecule. So we have seen that uh, the thick filament and thin filament in A band, how they are arranged. So we have seen that for every thick filament, there is surrounding six thin filament, which is arranged in a proper hexagonal pattern. So this arrangement of a thick and thin filament is somewhat due to because of titin. Plus, they are made of filamentous molecules, so they are act as a molecular spring, which add elasticity to the muscle. Then there's another molecule we call them as actinine, and the function of actinine is that they anchor the thin filament with Z line. So thin filament are attached with the Z line. We have seen that the actin is filament is attached with the Z line because of a molecule which we call them as actinine. Desmin, desmin is the molecule which attaches the Z line to the plasma membrane. Okay. So this attachment of Z line with the plasma membrane or sarcolemma of muscle fiber is because of a molecule which we call them as desmin. So titin, actinine, and desmin. Now coming to an important molecule which we call them as dystrophin, because mutation at this level on this molecule causes you can say uh, disease which we call them as muscular dystrophy. So muscular dystrophy is because of many reasons, and one of the congenital causes of muscular dystrophy is because of mutation in the gene for the protein, which we call them as dystrophin. So whenever the gene which is responsible for the formation of dystrophin molecule is mutated, so that and then they can produce a muscular dystrophy disorder or muscular dystrophy. Got it? So here, what is the first you should understand what is the role of dystrophin. So in this diagram, you can see the dystrophins are you can see like a rod-like structure. Okay? They're made up of protein. This dystrophin, they are large dystrophin molecules, and this the function of this dystrophin molecule is that they attaches the thick the actin filament to a transmembrane protein, which we call them as beta dystroglycan. Got it? So it Uh, I think audio has been restored. The voice is coming. Okay, I will discuss. So just the last two slides are there. So what I was discussing is that about muscular dystrophy, about protein which we call as dystrophin. So dystrophin is a, large protein which is connecting the actin filament to a transmembrane protein which we call them as beta dystroglycan. Now, whenever the gene which is forming this protein is being mutated, then there will be either no formation of this protein, or there will be very reduced or defective formation of this dystrophin protein, and it will lead to a disorder which we call them as muscular dystrophy. Now, the muscular dystrophy, because of mutation in the gene for dystrophin protein, are classified into two categories. One is your Duchenne's muscular dystrophy, and another is known as Baker muscular dystrophy. Both this Duchenne muscular dystrophy and Baker muscular dystrophy, both are X-linked disorder. X-linked means that the gene, the mutation is happening at the level of X chromosome. So it's more common in females. So in Duchenne muscular dystrophy, it's a serious type of dystrophy, and because of a mutation in the gene for this protein, there is a complete absence of dystrophin protein. So if there is a complete absence of dystrophin protein, then it will lead to Duchenne muscular dystrophy. Okay. But if there is a reduced amount of dystrophin, if there is a mutation, and if there is a, because of that mutation, there is a there is a formation of less amount of this protein, or there is a defective formation of dystrophin protein, then it leads to a milder form of disease, which we call them as Baker muscular dystrophy. Got it? So this Duchenne muscular dystrophy and Baker muscular dystrophy is because of a mutation in the gene for dystrophin protein. If there is a complete absence, it is Duchenne's, and if the mutation is such that altered type of dystrophin protein is formed, or a reduced amount of dystrophin protein is formed, then it's a Baker type of muscular dystrophy. Both are X-linked and common in female. Normally, the individuals with Duchenne muscular dystrophy, their survival is very less. Most of these kind of patient, they die before attaining their adulthood, and it is generally because of weakness in their respiratory muscles. So, because of this muscular dystrophy, they are not able to breathe well because there is a weakness in the intercostal muscles, which is responsible for ventilation. It's a very serious condition, Dushan's. Baker is the milder form of dystrophy. So with this, today we complete your neuromuscular junction and few things about the skeletal muscle, about the structural aspects of skeletal muscle, how the functional organization is there in skeletal muscles. Then the concept of T-tubule and the sarcotubular systems, and then how the myofibrils are there, and how thick and thin filament are there, and then dark band, light band, and then about sarcomeres. Okay, and what is the composition of your thick filament and thin filament. So remember, revise all these things, and when we will discuss in the next class, so we will see that how this interaction between thick and thin filament is happening, and because of that, the contraction of things are happening at the level of muscle is happening. So that is the topic that I think we will discuss in the next class. So we will end our lecture now. Is it okay?